I'm Jonathan Storkey. I work at Rothamsted Research in the UK. I'm a plant ecologist, and my contribution to the project has been to help build models of the plant distribution in the future in response to climate change. But what I'm going to do in the next 10 or 15 minutes is to give the headline messages, if you like, of the work that's been done on some of the environmental science around ragweed, and then I'm going to hand on to a colleague, Ian Lake, who will begin to talk to more about the consequences of human exposure and the clinical consequences of the spread of ragweed. So my first few slides are filling in a few of the gaps in terms of ragweed biology. So let's see if this is working. Okay, so there's the plant. You've already seen some photographs of it. Some key points to do with its biology and its physiology that are important for determining its range and its response to management. We've already heard that it's a summer annual, so it tends to emerge in the spring. It's a member of the daisy family, of the Asteraceae. A very important part of its biology is monoecious, which means that it has female and male flowers on the same plant. And that's very important because what we're interested in is the third photograph along there is the male flowers. That's the bit that's doing all the damage, that's producing all the pollen. So we're interested in seed production, yes, for the persistence of the plant, but we're also interested in the male flowers and how much pollen they're producing. It tends to require cold winters to break seed dormancy, so you need extremes of temperature for it to persist. And it's thought to have a northern limit to its range, which is determined by day length. It's killed by frost, so the flowering period and the pollen season will end as soon as you get the first frost. And we've already heard how pernicious this plant can be, that it can produce up to 3 billion pollen grains and very high numbers of seeds as well, and those seeds are very persistent. So it's a very successful alien plant. It's very well equipped to spread quickly. It's a good invader. This is where it originated, North America. It's a native of Canada and North America. And now has become, this is a map from the USDA page on ragweed with lots of background information on its biology as well. And it's now an invasive plant in Europe and also in China. It's a ruderal, an annual plant, so it needs disturbed land to spread and to persist. So the habitats you tend to find it in are those disturbed habitats, so annually cultivated crops, especially sunflower, along roads and railways, so it spreads along transportation networks. And importantly, in urban areas, where of course there's lots of people being exposed to the pollen, and also along rivers and waterways. This is a map that was produced by a recent project led by a colleague of mine, James Bullock, in the UK at the Centre for Ecology and Hydrology. And they drew together lots of national data on the current distribution of Ambrosia artemisifolia to produce this latest distribution map back in 2012. And the colours relate to the number of observations in the different cells. So the redder it is, the more often that plant was observed, and the green areas are where it's less frequent. You have to remember that this map will include an element of bias to do with sampling intensity, so areas where there's more sampling going on, you'd find more plants. But generally, it shows us the centres of distribution that Michel already mentioned around the Pannonian Plain and in central France. So first recorded in Europe in the mid-19th century and has spread rapidly after 1940, largely through contaminated crop seed and along transportation networks. And that's where the centres of distribution are, and that's reflected in where you find the most pollen. And we've already seen a nice animation of the pollen season, so I won't dwell on that. An important point to make is it's an ongoing invasion. And there's a couple of maps now from the literature from two very well-studied countries in France and Austria that show the stuff is still spreading. So even without climate change, the problem will get worse because it's still filling in available habitats in Europe. So what we've got here on the left, on the x-axis we've got date, 1900, 1950 and 2000, and then we've got the different departments in France and when ragweed was first observed in those departments. And we can see as time goes on, more and more, it occurs in more and more departments, even in recent history. So it's still spreading in France, and on the right there we see where it's mainly found with the intensity of the colour. And this is work by Bruno Cheval, who's done a lot of management work and a lot of work on the distribution of ambrosia in France. And you see a similar picture from Franz Essel's work in Austria. Here you have 4 maps. You have a time series. Top left is the earliest from 1883, and then it progresses top right, bottom left, and bottom right. And you can see, hopefully from where you're sitting, that the number of circles increases. The open circles are casual populations, and the closed circles are established populations. And another trend that they're seeing in Austria is populations that used to be casual, so they'll crop up for a couple of years and then die off. They're becoming established. So the problem is getting worse. So the approach that we've taken within the modelling group, Work Package 1 and Work Package 2, is to take a process-based approach to understanding the life cycle of the plant and use that to try and define its potential range under climate change. So we just take the life stages of the plant, whether it's in the seed bank, whether it's a seedling, a flowering plant or a mature plant, and we model the transitions between those states. And each transition will have an impact of weather and climate. It will be driven by weather and climate input and parameters. And when we do that, we can calculate how the population will change. So the numbers of seeds in year 1 divided by the number of seeds in the previous year. So it's giving you a population growth rate. And from that we can derive these maps that we use to define habitat suitability in terms of climatic thresholds. So the categories we've got here is U is unsuitable, U0 is very unsuitable, and U1 is unsuitable. Then we have C for casual and E for established. So as it gets more brown, those are the areas where we expect from the model output that the species will establish. And this is under baseline conditions. There's an element of uncertainty to this, of course, but it more or less maps onto current distribution. So we're confident we're along the right lines. So we can use that for current conditions, and we can also use it for future conditions, as you'll see in a moment. But just to make the first key message, that common ragweed has yet to fill its available physiological niche under current conditions. So even without climate change, we're saying that the problem will get worse. And now thinking about the effect of climate change, these are similar maps to the one I showed you before. So the darker brown it gets, the more the habitat is suitable for it to establish. We've got near future in the next couple of decades, and then the far future from the 2040 onwards. And you can see that we're predicting a northwards spread of that top limit of its range. So we're expecting the available physiological niche to increase with climate change as the climate gets warmer. So these maps tell us about the future physiological niche. Again, there's some uncertainty, but we're confident of that northward shift in range. And it corresponds to other papers in the literature that are saying the species will spread into West Europe especially. Okay, so that's just the effect of weather, that's just the effect of climate on its future distribution. But we've looked at lots of other factors that define how the extent and the magnitude of the problem. And there are other important factors. One is, are there available habitats for the species? So our models incorporate land use patterns. How fast is the species spreading? How quickly will it fill up that available niche? And that's a very important process parameter, and we look at different scenarios that Ian will refer to in his talk. If we assume it just stops and it doesn't go any further, what will happen to the problem? A slow and a reference and a fast spread. We've looked at how will climate affect within season pollen production and emission. Sorry, there's a typo there. It should say within season pollen production in terms of how big does the plant get and how much pollen does it produce. In future climates, will we get bigger individual plants and more pollen? What's the length of the pollen season? So will they flower for longer and therefore produce more pollen? And what are the daily release patterns for pollen release and transport, and will that change under future climates? So we have incorporated all of those things in our models. We've looked at it across different temporal and spatial scales, and we now have data that means we can predict within season pollen emission in terms of total European load of ambrosia pollen from seasonal weather patterns, and also quantify the relative importance of those different drivers in terms of determining the overall pollen load in the future. So we've not only looked at physiological thresholds and the distribution, we've also looked at within season processes and dynamics. And the key message from all of that work is that climate change will lengthen the pollen season, increase pollen production per plant, and extend its distribution. So climate change will increase the magnitude of the future problem and its geographical extent. And then a final slide to pick up on some of the things that Michelle was referring to in terms of looking at allergenicity of the pollen. We've not only looked at the pollen in the atmosphere, we've also looked at atmospheric pollutants using atmospheric pollution and transport models to look at the interaction with the pollen and predict the effect of ragweed allergenicity. We've used a mouse model to look at that in the lab and found a dose response of allergenicity to exposure to ragweed pollen, and also differences between where that pollen comes from. So urban populations were more allergic to the mice than rural populations. So it suggests there's some interaction with the environment going on, although it's been difficult to manipulate that in vitro. So my final slide and my final key message is airborne pollen cannot be ignored in seeking to improve air quality, and its inherent health impacts and interaction with air pollutants also needs to be considered. And with that, I'm going to hand over to Ian, who will talk to you about the consequences for human exposure.
Thank you, Jonathan.
Okay, thank you, Jonathan. So, my name's Ian Lake, and I'm from the School of Environmental Sciences at the University of East Anglia in the UK. And I guess my fascination with Atopica is really trying to understand what the impacts of climate change will be upon the health of the population. And I think one of the starting points is if you look at the fifth assessment report of the IPCC and the section that talks about what the impacts of climate change will be on allergic disease, there's actually very little there. There are a few papers that make some suggestions as to what the impacts will be, but no studies that start off with the climate and work right the way through to impacts on population. And I think that's the real gap that Atopica is actually filling within the academic literature and in the policy literature as well. So that's what we're trying to do, go from the climate right the way through to the impacts on pollen. So what I'd like to do is take a slightly different tack to Jonathan and start off with my key messages, discuss with you how I've come up with those, and then repeat those at the end. So the first key message they're going to be presenting to you is that the number of individuals at the European level with allergy symptoms related to ambrosia we think is likely to more than double from current levels to when we get to the far future by 2041 to 2060. 2/3 of those changes are driven by climate change, but as Jonathan was saying, ambrosia is spreading in any case, so we have a climate component and we have another component which is just that ragweed is spreading. Most of the increase in allergy symptoms in people with allergy symptoms that we're seeing are going to be in northern and eastern Europe and also northern Italy as well. And finally, I'm going to show why we think the control of ambrosia has really important public health benefits. In order to do that, what we need to do is generate what we term a dose-response curve. So to say, for differing levels of pollen, what will the response of the population actually be? And And looking at ambrosia pollen is quite different to other areas of air pollution, such as particulate matter or ozone, where there are well-published dose-response curves that you can use for these kind of studies that tell you if you have a small increase in PM10, you get a certain response in the population. We don't have that for pollen. So what we started off doing is by doing a systematic review, so a structured review of the academic literature, searching for all studies at the European level that have basically report a sensitization level, so the number of people who are exhibiting symptoms related to ambrosia. So looking through the literature, we came up with 30 such studies across the whole of Europe, over 20 different European countries. And because some of those studies report data for multiple parts of countries or over multiple countries, we ended up with 74 estimates of the number of people sensitized to ambrosia across the whole of Europe. Using some of the pollen models that were produced, we then effectively plotted up the sensitization rate against the level of pollen to generate a dose-response curve that looks something like this. So the blue line is the dose-response curve, and we have what we would term a logarithmic relationship. So right at the bottom of that curve, for example, around levels of 2,000, an annual load of pollen of around 2,000 grains per metre squared, we would expect to see a sensitization of around 20%. And as we go to higher levels, the percentage of the population sensitized increases, but at a lower level. So by 6,000, it goes up to about 25%, and by about 12,000, it starts to really reach a plateau at about 30%. So once you have that dose-response relationship, it's then a moderately straightforward operation to then take the pollen models that are produced and map them onto human health consequences. So from the maps for grid squares across Europe, we had a pollen level. We also had population data for each of those pollen levels, and we've done everything at the NUTS2 level. We can then take the pollen level, estimate the percentage of the population sensitized, and multiply that by the population to start coming up with numbers that are important to public health officials. Now, I guess to give you a sense of the complexity of some of the work that's gone on within Atopica, we have a whole series of different layers of results. And what I'm going to do is explain the results we have, and also just to say what I'm going to focus on. So in terms of thinking about climate change, about effects into the future, we have 3 time periods. We have our baseline time period, we have our near future period, which we're terming 2021 to 2040, and we have our far future period, which we're terming 2041 to 2060. For each of those, we have 2 indications of the amount of climate change using the terminology, the representative concentration pathway. And that gives us an indication of the amount of carbon dioxide in the atmosphere. So 4.5 would be a moderate level of carbon dioxide in the atmosphere, and if emissions aren't controlled, then we might see the emission pathway being higher, so we might see a higher concentration of CO2. We did actually find that there was not actually, in terms of the results that I'm going to show, much difference between those 2. So I'm simply, for simplicity, going to present to you the results for RCP 8.5 for a higher level of CO2 in the atmosphere. The results for 4.5 look very similar. One of the things in terms of climate change that are really key to understand is the uncertainties in all the climate modelling. So Atopica has been done with 2 different regional climate models. One with RegCM and another one CHIMAIR. And looking at those 2 gives us an indication of the uncertainties that we may have in our data. And what I'm going to do again for this presentation is give you the average of those 2. If you come along to the presentation tomorrow, we'll be looking in a little bit more detail about where some of these models are diverging slightly more in terms of their predictions of populations at risk. And then finally, we have the different invasion scenarios that Jonathan was talking about. So the first is where we're holding ambrosia where it currently is, we're not letting it spread. We know that would be almost impossible to achieve. We then have a slow invasion scenario. We then have a reference scenario, which we think is the most likely one, and we then have our rapid one, where controls on ambrosia would be, well, be a lot stringent than they are. So if you simply sum those numbers, the green line there gives you an indication of how the change in allergy symptoms related to ambrosia we think will change into the future. So at the moment, we're estimating, based on the sort of baseline models, around 25 million people in Europe sensitized. That's expected to increase to above 30 by 2021 to 2040, and then to over 50, so more than doubling by 2041 to 2060. But also, what's probably almost more important than that is if you look at the impact of the different invasion scenarios, and you see a big divergence from that green line. Sorry, from the green line in the middle. So if we reduce controls on ambrosia, if we have what we term the rapid invasion scenario, the numbers of people sensitized would be far greater, up to about 75 million by 2041 to 2040. If we have much stringent controls, we have a slow invasion, those numbers are slightly lower, up to about 30 million. But even if we don't allow ambrosia to spread any more from where it currently is, we would still see increases in the order of several million individuals. So just to summarize, sort of repeat those key messages, so we think the number of individuals with allergy symptoms related to ambrosia is likely to more than double from current levels by 2021 to 2040. 2/3 of those changes are driven by climate change. We have to recognize that ambrosia is spreading in any case. Most of the increase that we observe is happening in northern and eastern Europe, and also northern Italy as well. And finally, control of ambrosia has important public health benefits. If we allow ambrosia to spread more than it currently is, or we really halt the spread, we can have very big impacts upon the health of the population.
Thank you, Jan.